Imagine being told that every standard treatment that you've tried, chemotherapy, radiation, and even a stem cell transplant, just isn't working. That was a reality for Emily, a 6-year-old girl with relapsed leukemia. With nowhere left to turn, her parents and even her doctors had run out of options. And yet, they made one last call, an experimental therapy that sounded almost futuristic, one that would reprogram Emily's own immune cells to fight her cancer. Within weeks of receiving the treatment, something extraordinary happened. Emily's cancer disappeared. And today, more than a decade later, she's still in remission as the first child to receive CAR T-cell therapy. Emily's recovery was the first piece of evidence that this radical therapy could actually work. But what exactly happened inside her body? Welcome back to the Cells and Stories podcast. I'm your host, Khadija Dundia, and today we're exploring how CAR T-cell therapy is reshaping the fight against cancer. To understand CAR T-cells, you first need to understand what normal T-cells are built to do. T-cells are a part of the adaptive immune system, which is the specialized branch of immunity that learns and remembers how to respond to specific threats. You can think of them as highly trained security officers. They don't attack anything that looks suspicious. Instead, they are highly specific. They look for evidence, for a clear signal that the cell is infected or gone rogue. But what do T cells use as evidence? Well, every cell in the body displays ID badges, called MHC, on its surface. But these ID badges aren't just labels, like your name and address on your driver's license. MHC can also give T cells snapshots of what is happening inside the cell. If the cell is healthy, the badge shows T cells harmless proteins. But if the cell becomes cancerous or infected with a virus, MHC presents abnormal fragments, like mutated proteins or pieces of the virus itself. Fragments that clearly don't belong in the cell. T cells are constantly scanning these badges. They do so by using a receptor on its surface, called a T cell receptor. T cell receptors can only recognize the shape of certain fragments, kind of like a key matching a lock. But when the T cell receptor finds a cell with the right match, it binds right to it. Once it's locked in, the T cell releases toxic molecules that kill the diseased cell and signals other immune cells to join the attack. That's exactly the job of T cells, to identify and kill diseased cells. And although T cells are incredibly powerful, they are not perfect. In fact, cancer exploits a loophole in this very system. Because cancer cells are a mutated version of your own cells, its MHC ID badge looks normal to T cells. Cancer cells may even reduce the number of MHC molecules on its surface, so that T cells can't scan them at all. This means that cancer cells can slip up under the immune system's radar. Interestingly, CAR T therapy helps resolve this very issue. Here's how it works. Firstly, doctors draw the patient's blood and separate out the T cells. The T cells are taken to a lab where they are genetically engineered to produce special proteins on their surface. These proteins are called chimeric antigen receptors, or CARs. CARs supercharge the T cells, helping them to better latch onto and kill cancer cells. After the T cells have been modified, they continue to grow and multiply in the lab. Eventually, these T cells are finally ready to be infused back into the body through an IV. Once in the bloodstream, CAR T cells will continue to grow. And if all goes well, the supercharged T cells will be better able to kill off cancerous cells and ultimately eliminate the disease. It's clear that CAR T cell therapy is a powerful and groundbreaking strategy to fight cancer, but this power comes with its trade-offs. One of the largest challenges is a phenomenon known as cytokine release syndrome, or CRS. When CAR T cells encounter their target, they don't just kill the cancer cells. They become extremely active. They start pumping out cytokines, chemical messengers that immune cells use to coordinate an attack. A balanced, moderate amount of cytokines is exactly what you want because it means that the therapy is working. But when a large number of CAR T become active all at once in the body, cytokine levels can spike to dangerously high levels. These cytokines recruit even more immune cells, which then release even more cytokines. This loop of cytokine creation can lead to a cytokine storm, where the patient begins to experience physical effects. Symptoms can include high fevers, dangerously low blood pressure, rapid heart rate, difficulty breathing, and damage to organs like the liver and kidney. In the most severe cases, doctors intervene with a medication called tocilizumab, which blocks IL-6, one of the central cytokines responsible for driving this inflammatory storm. Another complication affects the nervous system and is known as immune effector cell-associated neurotoxicity syndrome, or ICANS. Researchers believe this happens when high levels of cytokines make the blood vessels in the brain temporarily leaky. This allows immune cells to enter areas they normally shouldn't. Patients might become confused, have trouble speaking clearly, experience tremors, or, in some rare cases, develop seizures. Currently, it's still not fully understood why some patients develop only mild symptoms while others can experience life-threatening neurological effects. The biology behind ICANS remains one of the most active areas of CAR T research. And then there's the issue of the cancer cells themselves. Cancer cells evolve constantly. Some cancer cells manage to survive through a process called antigen escape. During antigen escape, cancer cells stop producing the target that CAR T cells are meant to recognize. Other cancer cells release suppressive signals that reduce CAR T cell activity, or even express molecules that can interfere with the survival of CAR T cells. Beyond the biology, there are also logistical hurdles. CAR T therapy is made from the patient's own cells, and producing it takes time, sometimes upwards of 2 to 4 weeks. This is because the cells have to be collected, genetically modified, multiplied, and quality checked. For patients with quickly progressing disease, waiting this long can be risky. And because therapy requires specialized facilities and highly trained teams, access is limited, leaving many patients who could benefit from the therapy behind. So, those are the main limitations of current CAR T cell therapies. But now, let's talk about what researchers are doing to address them. One of the most prominent innovations are off-the-shelf CAR T cells. Right now, every treatment has to be custom-made from the patient's own T cells, which takes weeks and can cost hundreds of thousands of dollars. Off-the-shelf approaches try to change that entirely. Instead of waiting for a patient's cells to be collected, engineered, and multiplied, scientists can take T cells from healthy donors, modify them in advance, and have them ready to use. Another major innovation is the development of dual-target CARs. Earlier, we talked about antigen escape, when the cancer cell changes or loses the molecule that CAR is engineered to target. Dual targeting tries to address the problem of antigen escape. As a part of this approach, scientists engineer CAR T cells to recognize 2 different molecules at the same time, rather than just one. This makes it harder for the cancer cell to hide from the CAR T cells, making the CAR T cells more effective at their job. Furthermore, scientists are also building safety switch systems directly into the cells themselves. Some designs include suicide genes that can be activated if the reaction becomes too intense. Others use small molecule switches, which are essentially an on and off button that doctors can control. These tools give scientists more control over CAR T cells. But T cells aren't the only ones that can be engineered. In fact, natural killer cells, or NK cells, are being turned into CAR NK cells. Macrophages, another kind of immune cell, are also being reprogrammed. Some labs are developing CAR regulatory T cells, or CAR Tregs, not to fight cancer, but to dampen the immune system in conditions like autoimmune disease and organ transplant rejection. None of these approaches is a complete solution, and many are still years from routine clinical use. But they do highlight how the field is expanding, not just in terms of improving current CAR T cell therapies, but also broadening what engineered cells can do. As these strategies develop, they have the potential to extend the benefits of cell therapy beyond cancer and toward other complex conditions, marking the next stage and the evolution of immunotherapy. CAR T therapy began as the only remaining treatment option for a single patient named Emily. Now, over a decade later, it has become one of the most significant advances in cancer treatment. But as we've covered, the current versions of CAR T have limits. They can trigger intense immune reactions, they don't work for every patient, and they remain difficult and slow to manufacture. And cancer itself continually adapts, forcing researchers to rethink their strategies. The work happening now is aimed at addressing each of these barriers. Off-the-shelf CAR T cells seek to reduce the wait time of traditional CAR T cells. Dual target designs prevent antigen escape. Safety switches make treatment more controllable. And newer engineered cell types, like CAR NK cells and CAR macrophages, are being studied for their ability to treat solid tumors or non-cancer diseases. While there is more progress necessary, together, these innovations in CAR T cell therapy show where the field is heading. We're moving from a single therapy toward an entire toolkit of engineered immune cells, each designed for a different challenge. As these innovations develop, they're likely to broaden who can receive cell therapy, how quickly it can be delivered, and what kind of diseases it can treat. That brings us to the end of this episode. Thanks for listening to Cells and Stories. I'm Khadija Dundia, and I'll talk to you next time.